translated into protein. Okay, let's see. Okay, this is your DNA. Okay, so we know that DNA, this is the nucleotide of DNA. Okay, cytosine, thymine, guanine, adenine, and when it transferred into RNA, okay, we know that in 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 RNA there is no thymine, so it's going to be transferred to the cell. Okay, then it's going to be translated into protein. Okay, so this ETD refers to this is the initial, okay, the initial name of the specific protein. Okay, E is stand for one kind of protein, and symbol for T and J. Okay, all right, so this is this information. Okay, so information in nucleic acids can be replicated or transcribed, so the information flow is reversible. So it is a reversible process. Okay. However, there is no flow of information from protein back to RNA or DNA. So it's a reversible from RNA to DNA. Okay. So replication and transcription is a reversible process. Okay. So these are the basic rules of DNA replication. So there are 5 basic rules of DNA replication. So DNA replication is semi-conservative, okay, and replication begins at a defined origin. Replication process bidirectionally, and replicative process is in 5' to 3' direction. Is a process of semi-discontinuous. So basically, we have to understand about all of these terms. Okay, it's mean by semi-conservative, defined origin bidirectionally, 5' to 3' directions, and semi-discontinuous. Okay, these are the basic rules of the replication. Okay, so these are the basic requirements. Okay, tadi basic rules. Now it's the basic requirements of the replication. So for the replication to be conducted, so we need all this. We need a primer. Okay, what is primer? So primer is DNA it helps DNA polymerase to initiate DNA synthesis. So basically, I can say that if you want, so these are the basic requirements of DNA replication. Let's see if you want to run PCR. So you need to have all this. Okay, we need to design our primer. We need to have enzymes. We need to have this concentration of this nucleotide. We need enzymes. So we need this replication. Okay, so these are the basic requirements of DNA replication. Okay, so replication begins at a defined origin. Okay, so basically, okay, in chromosome, okay, it's normally for bacterial yeast chromosome, it appears as a circular, okay, as a circular chromosome. It's an adenine. Okay. For human chromosome, basically it more appears as a linear chromosome, but for bacteria, it appears as a circular, okay, circular chromosome, and when it under microscope, it can be seen there's a bubble, there's an I chromosome, okay, dia macam ada lamp ke atas tu, okay, so that represents as a defined origin. So defined origin is a place where the replication occurs. Okay. So in this diagram, it gives you so many information basically. So this is the origin replication. Okay, this is the origin replication, and at this it started, the replication started here and it began to run bidirectionally, okay, two different directions, and it's also known as a replication fork. Okay, replication fork means it is another term, okay, another term to show that dia macam apa, macam fork kan, okay, okay. So this how, okay, from double stranded DNA, okay, to start DNA replication, it needs to be unwind. Okay, it needs to be unwind into linear strand of DNA. Okay, okay. Okay, so this is what I mentioned just now under microscope. This is how the circular chromosome looks like. It looks like as a bubble, okay, or it's called as an I chromosome because this structure looks like a bubble structure. So this is the origin, okay, origin or known as a ORIC, okay, origin of chromosome, okay, okay. So for eukaryotic chromosome, okay, eukaryotic chromosome will have multiple origins of replication, That's why it also once it runs bidirectionally. It runs bidirectionally and it will have replication fork. So that's why it's called as a fork, because kalau in eukaryotic chromosome it has this multiple origins of replication, so it shows that it will have multiple replication fork as well. Okay, multiple replication fork. So that's what it looks like macam fork. So hence it is named, okay, the term is named as a replication fork. Okay. Okay, so this is an example of eukaryotic chromosome. It may have more than one origin of replication. You all this soalan ke? Okay. Okay. So only fully methylated origins can initiate replication. What does it mean by methylated? Okay. Do you know what is methyl group? Do you know what is methyl group? Anyone? What is methyl group?
CH3.
What is methyl group? Anyone knows what the structure of methyl?
CH3.
Okay, methyl group is CH3. Okay, when you see CH3, it's also known as a methyl. Okay, methyl group. So you can see that this is an enzyme. It's an enzyme methylase. So this is enzyme that add methyl group to the structure of DNA. Okay, this then methylase that methylate. Okay, this enzyme methylate this DNA by adding methyl group, which is CH3. Okay, CH3. Okay, so basically in specific, this enzyme add this methyl group to adenine. Okay, kita ada banyak kan, in DNA we have these 4 nucleotides. Okay, adenine, thymine, guanine, and cytosine. So in specific version, this enzyme add this methyl group to adenine. Okay, to adenine. Okay, to adenine, so it activate. Okay, so it activate the adenosine. So it activate this the origin of DNA. So it can start the process of replication. Okay, so when you see CH3 is a methyl group. Okay, dia banyak tau. Dia banyak penggantian. Okay, methyl group is CH3. Okay, so okay, you can see this is methylated DNA. So let's say we have this nucleotide in DNA strand. We have G, we have A, we have B and C. And that enzyme then methylates is going to add the methyl group at adenine, specifically to adenine. Okay, dia tak letak, dia tak add dekat thymine ke cytosine ke, it's specifically for adenine. Okay, the methylated process. Okay, that's methyl to CH3. Okay, so in DNA synthesis, okay, it is run in 5' to 3' direction, and it's also known as the semi-discontinuous. Okay, what does it mean by semi-discontinuous, and what does it mean by 5' to 3' direction? Okay, let's see. We have this, this is a double stranded DNA. Okay, double stranded DNA. So basically, it should started with the unwind of DNA. Okay, it should started with, tapi saya letak kat belakang lah. Sebab saya nak introduce dulu what does it mean by the semi-discontinuous. Okay, so once the double stranded DNA unwind is unwind, okay, we separate the double stranded DNA into two single stranded DNA. So we know that DNA runs from 5' to 3' direction. So the the strand at 5', okay, is act as a leading strand. Okay, act as a leading strand, and another strand at 3', okay, act as a lagging strand. Okay, so what does it mean? What is the difference between these two strands? Okay, the leading strand, it can be synthesized discontinuously. Okay, meaning that new peptides can be added. Okay, continuously. Okay, I have discussed this during last lecture. Okay, how DNA being incorporated into each other to become one, to become double helix, because 5' of phosphate is combined with 3' hydroxy group of another DNA. So it means that nucleotides being added continuously to the 3' end of DNA. Okay, that's why this leading strand, it can be synthesized continuously. Okay, but for lagging strand, it cannot be synthesized. It is synthesized discontinuously because we need new primers. We need primers that can add new nucleotide. Okay, that can add new primers that helps to add nucleotides into the DNA strand. Okay, ada masalah tak? Okay, now you have this semi-discontinuous and also 5' to 5' direction, and you also have this lagging and leading strand. Okay. Okay. So we know that DNA synthesis is in the 5' to 3' direction. Okay, 5' to 3' direction. There are semi-discontinuous and prime by RNA. So prime by RNA meaning that you need to have primer. Okay, what is primer? Primer is a short nucleotide. Okay, short nucleotide that helps to initiate the process of replication. Okay. Okay. So on the leading strand, DNA synthesis can proceed continuously, but on the lagging strand, the process is occurs discontinuously. So that's why it's called as semi-discontinuous, because we have the leading and lagging strand that one is occurs continuously, another one is occurs discontinuously. Okay. Let's say, okay, this is 5', this is your double stranded DNA. It needs to be separated, so this is your 5', strand, and the upper one is 3' strand of DNA. Okay, so the 5' strand act as a leading strand. Okay, so the leading strand, okay, the lagging strand on the upper strand will act as a lagging strand. So this lagging strand needs primer. Okay, needs primer. Okay, needs primer to helps in the replication process. Okay, now, okay, so primase will synthesize RNA. So primase is an enzyme that synthesize primer. Okay, primase is the enzyme to synthesize primer. Specific kan? Primase and primer. Okay, then once we have this primer, okay, primer cannot act alone. Okay, we need to have DNA polymerase enzyme. So what does this enzyme do? So this DNA polymerase enzyme helps to expand the RNA primer into a positive fragment. Okay, so now we have this primer. Okay, so we have this primer. So DNA polymerase. Okay, what does it mean by DNA polymerase? So it extends. Okay, we have this primer. It extend the primer into a very quite not that long sequence of DNA, which called as a positive fragment. Okay, so now we have this positive fragment synthesized. So they are going to use this positive fragment to replace the RNA primer and to continue the DNA replication. So you can see there is a gap between the first initiated positive fragment with the synthesized one. So this gap is going to be sealed by one enzyme called as a ligase. Ligase, it helps to close the seal. It helps to 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 combine two different DNA to combine it together, so it becomes one complete DNA strand. Banyak kan? So you have primer, you have primase, you have DNA polymerase, you have ligase dekat sini. Okay. Okay. So basically, the process can be divided into three important parts: initiation, okay, elongation, and also termination. So initiation involves recognition of the position on a DNA molecule where the replication will begin. Okay, initiation, okay. So that's why we have this origin of replication. So it needs to be recognized for the replication to begin. The elimination, it started at the bidirectional direction at the replication fork, where the parent polynucleotides are copied, and the termination it occurs when the parent molecule has been completely replicated. So basically, once the replication process complete, is going to be terminated. So we don't want it to replicate unnecessary sequence. Okay. Okay, so during DNA replication, each DNA strand is used as a template to synthesize the second DNA strand. Okay, so DNA strand is always synthesized in the 5' to 3' direction. Okay, so this is your DNA strand. Okay, well, the first thing first, it needs to be separated. Okay, it needs to be separated into two single strand of DNA. Okay, two single strand of DNA. And these two single strands will act as a template. That's why you are the leading strand, you are the lagging strand. Both adalah template to synthesize the second DNA strand. Okay. 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 Another term that you need to know. Okay. Theoretically, DNA could serve as its own template in one of three different ways. There are three different ways of replication. Okay, we have semi-conservative, we have conservative, and also we have dispersive replication. Sorry, semi-conservative replication, conservative replication, and also dispersive replication. So what is semi-conservative replication? So semi-conservative replication so it uses each strand as a template. Okay, each strand as a template for a new strand. Okay, so each new DNA double helix would then have one parent strand and one new strand. Okay, this is your double helix. Okay, it's going to be separated and act as a template. Okay, okay, the first one acts as the first template, and the second strand act as a template. So it's going to be synthesized new strand. Okay, to generate one conjugate strand. So for semi-conservative application, we have one new parent strand and one new strand. Okay, we will have one parent strand and one new strand. Okay, satu parent, satu new. This is semi-conservative replication. Okay, so contoh ni lah sama juga lah. So this is the parent, this is the parental DNA double helix. This is the parent strand. So the red strand is a new strand. So this is the first cycle. Okay. So the second cycle you will have this. Okay, from 1 you replicate into 2, from 2 replicate to 4, 4 then replicate into 8. Okay, kalau 8 lepas tu 16, lepas 16 you will have 64. Jangan mistaken, 64. Okay, you gantikan. Alright. So this is how DNA replication of the soil. This parent strand will act as a template. Ini semua akan jadi template kat sini. Okay, kalau kat sini semua ni akan jadi template. So that's why from 8 it can jadi 16. Okay. Alright. For conservative replication, it's going the original DNA will remain intact. So DNA molecules will be generated new after one round of replication. So there will be no mixing of, there will be no parent strand in new strand of DNA. Okay, for this specific replication, it have it contain the fragments. Okay, the fragments of parent DNA. Okay, into this newly strand of DNA. Dia ada mixture of fragments. Okay, so these are the researcher that prove that the DNA replication is semi-conservative, which is Nicholson and Starr. Okay, they conducted an experiment by using 50 nitrogen. Okay, to confirm that DNA undergo the process of replication is by semi-conservative. Okay, I have shared the detailed video of this Nicholson and Starr experiment, so you can have a look. Okay, try to understand that. It's a very straightforward. It's an easy to understand video. Okay, so I shared that. Please have a look at the video. Okay, after this. Okay, so we know there was an anchored proposed that DNA is double helix. Okay, it has two strands that separated from each other, but it remains intact as each serves as a template. Okay, so this theory has been proved by this experiment. Okay, you can have a look at this YouTube video that I have shared in the class. Okay, so basically, the experiment used this nitrogen 15. We know that the most abundant nitrogen 15 is this nitrogen. Okay, that they labeled the bacteria with this this heavy nitrogen, which is nitrogen 50. Then once it labeled the nitrogen is nitrogen 15, then it transfers into this the most abundant nitrogen, which is nitrogen 14. Then it's centrifuge them. It's try to centrifuge and look at the density. So from here they can see that the replication process. The replication process occurs as a semi-conservative. Bila datang, okay, set lah. Okay, okay, alright. So, okay, this is how the DNA, the replication process occurs, which we have this DNA strand that is separated. So we have this leading and lagging strand. Okay, at 5' strand is a leading strand. So this leading strand can be synthesized continuously, but for lagging strand, it needs primer that can be extended into oxidative fragment by DNA polymerase. Okay, you all kalau tak faham tanya, okay. Okay, so these are the 4 requirements for semi-conservative replication. Okay, DNA must act as a template for complementary bestowing. Okay, we know that. Okay, we know that. Okay, each strand will serve as a template. Okay, and this nucleotide, okay, the ATP, the GTP, the CTP, and the TP must be present. So basically, this helps the process of replication. Okay, kalau kita, if we conduct a PCR experiment, we require this. Okay, kalau ni dalam bentuk DNTP lah. Okay, I'm using DNTP okay, for research. Then we need to have DNA polymerase enzyme. Okay, DNA polymerase enzyme is a very important enzyme that is responsible to add the nucleotides to extend the DNA strand. Okay, it helps to bring the substrates to the template and catalyze the reactions. Kalau tak ada ni, if you have the primase, if you have this, okay, without the DNA polymerase, the replication process will not occur because this is the enzyme that add nucleotides to your DNA strand. Okay. Okay, we need to know this. The reactions involved in replications are endogenic. So there are source of chemical energy must be present. Okay, require energy. Okay, now kita baru nak start. Okay, then replication takes place in 2 steps. Okay, the hydrogen bonds between the 2 strands are broken. Nampak ini yang saya kata tu. Okay. We know that the hydrogen bonds that hold, okay, between the two nucleotides, okay, that makes to the double helix, kan? Okay, so these hydrogen bonds will be denatured, okay, will be broken down, so to separate the 2 double helix into 1 single strand. Okay, so one single strand dia ready kan DNA strand tu for nucleotides to complement, okay, to new nucleotides come in and complement, okay, to become one complete DNA. Okay, then the nucleotides are covalently bonded to each bonding strand, and in virtually ordinary application, nucleotides are added to the 3' end of the bonding system. Okay, the addition of nucleotides, okay, at the leading strands, it will be added at the 3' end. Okay, it will be added at the 3' end of the bonding nucleotide. Okay. Okay, so what happened during the extension? Okay, so basically this hydroxyl group at the 3' end it acts as a nucleophilic, okay, which is going to attach the phosphate group, okay, at the 5' end of another nucleotide, okay, to start the expansion process. So the hydroxy group, it attacks the phosphate group. It acts as a nucleophilic. It attacks the phosphate group at the 5' end of another nucleotide to begin the extension process. Okay, to start the extension of DNA. Okay, so the three phosphate groups of the deoxyribonucleic triphosphate are attached to the 5' position of the sugar. So it attacks the phosphate group. So where does the energy come from? So the energy comes from the breaking from the breaking bonds, okay, between these three phosphates. We know that in phosphates, in the deoxyribonucleic triphosphate, we have this triphosphate. We have three phosphate molecules. So by breaking down these three phosphates, it will it will release the energy. Okay, so it will supply the energy for the replication process. Okay, so the free hydroxyl group at the 3' end of the growing reaction reacts with one of the phosphate groups, breaking the bond between the phosphate group attached to the sugar and the two terminal phosphate group. So this is what happened, okay, during the extension process, okay, and the breaking of this one releases some energy synthesis, okay, by breaking of the phosphate, the two phosphate bonds, it's going to release an energy that can be used, okay, for the replication process. Okay, okay, this is the how the sugar phosphate backbone, okay, form. So the one phosphate group still attached to 5' carbon of the new nucleotide bonds to the 3' end of the growing chain. Okay. 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 So this is your this is your DNA. Okay. So when you have this new nucleotide, okay, this is your deoxyribonucleic triphosphate. So you have these three triphosphate. Okay. So by breaking the bonds between these phosphate, it's going to release an energy. So this hydroxyl group, the 3' hydroxyl group, okay, acts as a nucleophilic. It's going to attack this phosphate group, okay, of the other nucleotide, and is going to react together and join together to form a sugar phosphate backbone. So this how the addition of nucleotide occurs at the 3' end. Okay, so if that's what it means by this one. Okay, anybody? Okay, that number. Okay. Nucleotides are added to the 3' end of the growing nucleotide. Okay, the new nucleotide added at the 3' end. You can see added dekat sini. Yeah. Huh? So, yeah. Uh, can we have a break for 5 minutes, kejap lah? Please apologize. That's the challenge of working from home. Okay. Okay. So apa tadi ya? Ini saya belum tadi. Okay. So the addition of nucleotides, okay, occurs at the 3' end of hydroxyl group. Okay. And this is your deoxyribonucleic phosphate. So the breaking down of these two pyrophosphates will release some energy, and this hydroxyl group will attack this phosphate group of the new nucleotide to form a new sugar phosphate backbone, okay, of this double helix DNA, okay. Okay, which protein complex catalyzes the reaction of DNA replication? So we know that we have, okay, in circular chromosome there is an origin of replication, okay, and so we need this, okay, to start replication. It need to be recognized, okay, this origin of replication need to be recognized, okay, by the enzyme, by the complex of components, okay, that needed in the replication process. Okay, so we know that DNA replicates in both directions in bidirectional, okay, from the origin. So it forms these two replication blocks. So in the replication, both strands of DNA serve as a okay. So replication complexes meaning that these are the important component in replication process. So basically, these are the enzymes. Okay, paling penting enzymes. Okay, enzymes that involved in replication process. Okay, we have this helicase. We have this single-stranded DNA binding protein kinase. You have DNA polymerase 3 and 1, and also you have DNA ligase. So these are the enzymes. Okay. So helicase. So helicase. It denotes the double helix. So what does helicase do? It unwinds the double-stranded DNA. It separates the two double-stranded DNA. So there is a single strand binding protein that will keep the two strands separate. Okay, so then DNA helicase comes in. Okay, unwind the DNA. So there will be single-stranded binding protein as SBP that keeps the two strands separated. So once we have the free the linear single-stranded DNA. Okay, so now we need to have primer. Okay, we need to have primer to initiate. So RNA primase is an enzyme that makes a primer strand, okay, that serves as a starting point for replication. Okay, this one, okay, primase basically is an enzyme that yang ada dalam kita punya untuk buat primer kan, untuk to mix a primer strand. Okay, but let's say if you conduct, if we have research to we want to make a copy of DNA, kita nak replicate bacteria punya DNA, we want to run PCR, polymerase chain reaction, we have to design our own primer. Okay, we have to design our own primer, but it's not that difficult because there are so many software, there's so many apps that helps us to design the primer based on our specific sequence. Okay, macam during my time we are trained to design the primer manually, meaning that if you have that DNA sequence, we have to look, we need to identify at which sequence that we would like to amplify. So based on that sequence, we have to come up with our own primer. We have to design our own primer because primer is very important. Primer is very important. If you are using, if you are designing a primer that not accurate, that will lead to the mismatch of nucleotide, your DNA replication will not occur. You are not going to get any precise DNA sequence. Okay, so in our system, in our cell system, we have this RNA primase. So this RNA primase will design a primer that serves as a starting point for replication. Okay, so DNA polymerase adds complementary nucleotides to the protein strand, proofreads the DNA, and prepares it. Okay, then we have DNA like this. Okay, cells are breaks in the sugar phosphate backbone. Okay, you all okay, girl? Hmm? Hello. Irfan.
Ya, okey je.
Okay. Leading
Tengah
strand
dengar.
apa? What
Apa?
is leading strand?
The one yang apa tu, daripada yang first 5 ke 3.
Okay, so is the strand is a 5' strand that it make a leading strand and it can synthesize continuously. Okay, thank you.
Oh,
Alright, saja je. Okay, nak tengok you all ada ke tidak. Sebab saya tak nampak kan you all ni ada ke tidak kat dalam ni. Okay. Emma, where is Emma? Do you have Emma here?
Yes, doctor.
Emma, what is methyl group, Emma?
Sorry?
What is methyl group? What does this dimethylase do?
Dimethylase 2, is it?
Yeah. What is the role of dimethylase? Okay, what is methyl group? Go back to my first question. What is methyl group? Anyone? What is methyl group?
CH3.
CH3. Okay, so there is one enzyme, dimethylase. So what is the role of this enzyme? Fishatini? You have Fishatini?
Yes,
Okay, so what is the role of dimethylase?
doctor.
Sorry? Okay, anyone, what is the role of dimethylase? Add a methyl group to adenine. Okay, this phosphate to adenine of DNA. Okay, thank you. Saja nak ni. Saja nak apa? Nak break the silence. Okay. Alright, so basically this is, okay, we have this RNA complex, the RNA complex, the replication complexes, okay. So we need all these enzymes, okay, in the replication process. So this is the specific roles of each enzyme. Okay, we have helicase, we have the single strand binding protein, we have this primase, we have DNA polymerase and DNA ligase. So basically, these enzymes, all these enzymes, the role of each specific enzymes has it will follow their direction. So it started with helicase, so then single strand proteins comes in, then we have primase. Once we have primase, once we have primers, so DNA polymerase helps to add nucleotides, okay, to the growing strands. And if there is a gap between DNA in the growing strand, so then ligands help to close the gaps, okay, in the sugar phosphate backbone, okay. Okay, so let's look at the DNA polymerase. Okay, DNA polymerase cannot build a strand without having an existing strand of DNA or RNA called a primer. So primer is a very important pieces of DNA. Okay, primer. Okay, let's say kalau kita nak buat PCR, we need to design our own primer based on sequence. Let's say I would like to amplify gene, binding protein gene, for example, binding protein. Okay, I would like to amplify this binding protein gene. Okay, so based on the sequence, I need to design a primer. Okay, I need to design a primer. So it's very important. But in a biological system, we have that primase enzyme. Okay. So in DNA replication, the primase strand is a short strand of RNA complementary to the template strand. Okay, so it's a RNA complementary. Okay, so basically it's a short strand. You can, if let's say we need to synthesize our own primer, okay, primer, we can, the primer can be, we can design from the shortest maybe from 6 nucleotide, okay, up to 30 nucleotide probably, okay, depends, okay. Normally for my case, I would like to design from 15 to 25 long of primer, okay, 15 to 25 nucleotide primer. It depends on the purpose of that PCR process. Okay. So in biological system we have this primase to make the primer strand. So the primase is part of a protein complex called a primosome. Okay, and this primer is later degraded. Okay, it's later degraded and replaced with DNA. So the final DNA molecule will has no RNA because the ultimate goal for this process is we like to make a copy of DNA. Okay, so RNA is going to be degraded, okay, and replaced with DNA. Okay, let's see, we have this. This is the process of the DNA synthesis. Okay, once the double-stranded DNA has been separated, okay, then we have this primer, okay, that has been synthesized by primase. So once we have this primer, it helps to then DNA polymerase comes in and it added new nucleotide, okay, to the strand, okay, then it helps, okay, let's say this is A, T, G, C, so A, okay, so DNA polymerase helps to add T here, okay, because A is going to complement with thymine. Let's say in the leading strand we have G here, so RNA polymerase is going to add C, okay, because G is complement with cytosine. So that's the role of DNA polymerase, okay. So DNA polymerase finishes the DNA fragment. So when there is a gap, okay, gap between the DNA, the newly synthesized DNA, so so the old RNA primer is going to be degraded, it's going to be erased and replaced by DNA, and the gap between DNA is going to be closed by DNA ligase. So it's going to join the new DNA fragment to the closing chain. So it helps to close any any gap, okay, between the new DNA fragment. Okay, so this is primer. So it is a short RNA primer using a DNA template. So RNA primer. Okay, so it's a short strand of RNA. So basically, okay, boleh 18 nucleotide boleh. Okay, you can synthesize 30 nucleotide boleh. But there is in details, there is more characteristic to be followed, okay, in synthesizing RNA primer. Okay, banyak lagi dia punya karakteristik. How to design a good primer. Okay, first thing you need to make sure when the translation occurs, it's going to be translated in okay, DNA kan dia read by 3 nucleotide kan. Okay, contoh ATG, CAA, CGG, okay, dia tak baca AT. So this RNA primer need to make yang you design has to be able to translate to be read in 3 nucleotide lah. Okay, otherwise going to be mutation, it's going to be errors dalam ni. And also your primer need to have balance of GC and AT. But a good primer will have GC up to 50%. Why we need to calculate the percentage of GC dalam primer? Because we know that GC has 3 hydrogen bonds compared to AT. AT ada 2 kan hydrogen bonds. AT ada 2 hydrogen bonds. It shows that GC have stronger bonds compared to AT. That's why dalam primer, if we design new primer, we need to have at least the percentage of GC in the primer more than 50%. Okay, we would like to have a very good and strong primer. Okay. All right. So this basically this is a slide. This shows you on the direction of the frog movement. Okay, leading P mana leading to okay. It is bidirectionally. Okay, we have this replication fault. So it shows how the synthesis process occurs. Okay. So a little bit on the reaction, okay, of enzyme DNA helicase. It uses energy from ATP. Okay, to unwind the 2 DNA strands and make them available for complementary base pairing. Okay, so this single-stranded binding protein, it helps to keep the DNA strands to keep them separated. Okay, kalau small chromosomes and bacteria ada single origin of replication. For eukaryotes, okay, it can contain hundreds of origin of replication, and the process can occur at many different sites simultaneously. Okay. Ni. Okay, so this is the single strand DNA binding protein. So it stabilizes the separated DNA. It helps to keep the separated DNA keep them apart. Okay, to keep them separated. So it's going to bind at the strand once it's been unwind by helicase. Okay. Okay, so this is in general what happens during the replication process. Okay, you have this parent A, it being unwind by helicase, is being separated by helicase. So the single strand DNA binding protein comes in just to attach to the DNA strand to make sure the DNA strand keep separated. Then it comes, we have RNA primase attached to the DNA strand. It synthesizes new primer. So once we have primer here, it's going to be extended to a positive fragment by DNA polymerase, and it's going to add nucleotide, a new nucleotide to it, and it's going to be extended. Okay, then if there is any gaps between the new fragments of DNA, it's going to be closed by DNA DNA ligase. Okay, ligase enzyme helps to join two DNA fragments together. Okay. Okay. So basically, DNA polymerase ni, okay, we have okay. We have two DNA polymerase. We have DNA polymerase 1 and we have DNA polymerase 3. Okay, that helps to extend the new DNA strands. Okay. So, but in prokaryotes, DNA polymerase 3 is responsible for elimination of new strands. Okay. Remember just now I mentioned to you that DNA polymerase can help to repair any errors, okay, in DNA. So that one's the special ability basically okay. Okay, so basically DNA polymerase 1 so DNA polymerase 1 that responds, that has the ability to repair any errors in DNA. So basically, for DNA polymerase 3 is responsible for the elimination of new DNA strands. Okay. Okay, please recall that the new bases are always added to the 3' end of a growing DNA strand. Okay, and the strands in sample A are anti-parallel. Okay, all right. Okay. Okay, DNA synthesis occurs in 5' to 3' direction. Okay, so ini kita dah discuss tadi on the direction of the DNA synthesis. We have this leading and lagging strand. Dan sama juga lah, sama je. Okay, so this is another diagram that shows on the DNA replication process. Okay. Okay, now a little bit on the lagging strand. Okay, we know that leading strand can be synthesized continuously. Okay, for lagging strand, okay, it occurs in backward orientation. Okay, so it synthesizes discontinuously. So once we have RNA primase synthesized primer, so it's going, so DNA polymerase will come in and it's going to extend this RNA primer to synthesize the new DNA by working toward the previous oxidative fragment. So we extend the loop of the RNA primer to menjadi a very nice long oxidative fragments. Then this enzyme in polymerase helps to complement, okay, from the T prime end is going to add the new nucleotide to complement DNA, okay, towards the previous oxidative fragment. Okay. So DNA polymerase 1 then replaces the DNA primer of the previous oxidative fragment, then DNA ligase comes in to catalyze the formation of the phosphodiester linkages. Okay. Okay, so you need to take note on this. In A synthesis, the replication process, is occurs very accurate, very precisely. Okay, apa enzyme dia, apa reaction dia dekat mana, is a very accurate process. Okay, because if there is one mistake, if there is one error, if there is one mismatch in this pairing, will lead to origin of cases, will lead to cancer, will lead to so many other unknown uh, problem. Okay, sekarang banyak lagi yang discover. Okay, okay. Okay, now we move to DNA damage and repair. So, from previous lecture, okay, that's 1.5 hours. I hope you should be able to summarize on the replication process, on the basic rules of DNA replications, and what happens during the replication process from helicase to ligase. Okay, what are the enzymes that involved in the DNA replication? So what are the specific roles carried out by that specific enzymes? Okay, you need to understand the terms, okay, related in DNA replication. What does it mean by semi-discontinuous? What does it mean by semi-conservative? What is dispositive replication? So what roles carried out by helicase, by single strand binding protein, by DNA polymerase. Okay, so you need to know that. Okay, okay. So for DNA damage and repair, okay, so we know that DNA polymerase 1 can help to repair if there is any errors, okay, any mistakes, okay. What is the error rate? Okay, what is the rate of mistakes in our genetic DNA? Okay, in E. coli 1 in every 10 or of 10 base pairs inherited. So you can say it's very rare. But uh, if you are working with molecular biology, for example, if you are uh, try to replicate, okay, any DNA, if you don't have any, we know that replication process require this enzyme. We need DNA polymerase, we need this ligase, we need this helicase, kan, semua kan. Okay, let's say you are being given one FYP project to amplify apa-apa DNA. Okay, so you need to run PCR. Okay, you dah tahu tadi kan basic requirements, okay, basic requirements, basic what are needed. Okay, even though you have all that, okay, you need to design your own primer. You need to design. You need to have a polymerase. You need to have like this. But it's not that easy. Okay, because uh, certain different DNA will have different optimized requirement. Okay, for example, let's say you have designed a very good primer. Okay, but doesn't mean it's a very good primer until you run PCR. Sometimes you will not getting any DNA from that primer because maybe you have misplaced one nucleotide. Maybe you have mislook at once the translation occurs. Okay, what happened to the protein is going to be folded like this or it's going to be folded like that. Okay, or maybe the primer that you designed is going to produce when you sequence that DNA, you can see that there is a lot of mutation. A lot of mutation occurs. the the nucleotide has been replaced with other nucleotide. There is a mismatch here and there, so you are not going to get any DNA. Or maybe it requires low concentration of DNA. It maybe require high concentration of DNA polymerase. It depends. Okay, there is so many factors need to be optimized in order to get a very good DNA product. Okay. Okay, so DNA damage. Okay, there is a mismatch of nucleotide. There's a mutation that can lead to undesirable changes to genes. Okay, there are many factors that can lead to DNA damage. Okay, so why concern? Okay, what is important to why we need to know about the damage of DNA? Because if let's say exposure to mutagens results in initiation of tumors and other forms of cancer. Okay, do you have any idea what kind of mutagens that can lead to tumors? Any idea of what kind of mutagens? What are
Doctor, I cannot really hear you clear.
Oh, sorry. Sorry. Can you hear me clearly now?
Yes, I think it's better.
Okay. Okay, class. Do you have any idea what kind of mutagens that can, when you expose to it, it can result in this kind of that can lead to initiation of tumors or other forms of cancer? Contoh apa? Apa contoh?
Radiation.
Radiation, okay. Okay, this is a good example. Okay, radiation, what else? Chemicals, okay, contoh chemicals. Okay, once you enter the lab, okay, for your FIP or you are working in the lab during your internship, okay, be careful with certain chemicals, okay, because there are chemicals that when you expose it directly or bila terkena tangan ke or you inhale in a very significant of time can initiate this kind of problem. Okay, so please be careful. So you need to know the chemicals. Okay, there are chemicals that can initiate. Okay, that act as a mutagen. Okay, the radiation chemicals. Okay, there are so many things. Okay, X-ray, something. Okay, that's some sort of okay. So strong correlation between exposure to mutagens and cancer. So there are so many studies that has relate, okay, on the exposure of mutation, it can lead to cancer. So please be careful. That's why we have to be very careful dekat X-ray kan. So certain pregnant women cannot do any X-ray because it can cause any genetic modifications to the baby, for example. So every precaution that has been stated, so you need to really take it seriously. Okay, sama juga dalam lab. Okay, contoh I can give you one name of chemicals. Sama mesti apa nama dia? Ethidium bromide. Okay, ethidium bromide is one chemical that has been used in laboratory. So we use ethidium bromide to stain the walaupun guna kot ethidium bromide, saya tak sure dalam bila nak stain gel kan. Okay, when you want agarose stain, agarose gel, so agarose gel need to be stained with ethidium bromide. Okay, for us to visualize the gel. So the ethidium bromide, if you handle it wrongly, if you expose it to in a significant time, is a significant period of time, or is directly kena dekat you punya skin ke apa ke, it can, but it depends on that person. Okay, but please be very careful. Okay. Alright, so these are there are two types of replication errors, okay, either by substitution of the base pair, okay, or insertion extra base pair, or deletion. Okay, substitution, insertion, deletion. So this led to errors in replication. Okay, so the most common one is substitution. So it caused by polymorphism in the nitrogen base. So it leads to inaccurate bond pairing. Okay. Okay, this one is substitution. Okay, contoh substitution. Okay, let's say we have this, this is your sequence. Okay, but this base pairing between adenine and thymine has been substituted by GNC. Okay, or it can be this way. Okay, adenine and thymine, but it's going to be substituted with adenine here. It can be substituted to thymine. So even though only one pairing of nucleic, one pairing of base pair, but it can lead to the substitute of one base pair, and it can lead to many replication errors. Okay, disease lah, cancer lah. Okay, so this one is insertion. Let's say there's insertion of another base pair, so it is cause and replication errors. Same goes to the deletion. Okay, let's say one base pair has been deleted. Okay, it's a replication error. Okay, so there are two types of mutation, either it is spontaneous or induced mutation. So both mutations can lead to DNA damage. Okay, so what is spontaneous mutations? So basically it occurs spontaneously in a genetic trait. Okay, so either it is a part of a mismatch of the base pair or base modifications caused by hydrolytic reaction. So it can occur spontaneously. Okay, it can occur spontaneously. Okay. So how to detect and repair any DNA damage? Okay. So the first one is the cleavage of a phosphodiester bond holding the mismatched nucleotide. So we need to cleave, okay, the specific mismatch, okay, nucleotide by using the endonuclease, because it occurs inside kan kat dalam, so we use this endonuclease. Okay. Then we have to remove the mismatch base by using endonuclease enzyme. Then we can, then how to repair it, we incorporate the correct base pair by using DNA polymerase grade 1. Okay, grade 1 actually. Then this comes in to close the, if there is any gap, okay, to come up with the complete DNA strand. Okay. So these are the steps, okay, 4 steps, okay, how to do the repair. So we have endonuclease, exonuclease, DNA polymerase 1 to incorporate, and also ligase. Okay, so another common type of spontaneous damage is by hydrolytic reactions. Okay, for example, nucleotides containing purine bases can undergo spontaneous hydrolysis at the end glycosidic bond to remove the purine ring. Okay, it can happen. Okay, it can happen. Okay, this kind of reactions can be controlled because it's spontaneous. Okay. Also, heterocyclic bases can be deaminated by hydrolytic reactions. Okay. Okay, for example, conversion of cytosine to uracil. So, spontaneous damage is uncontrolled reaction, so we cannot do it, but it can be detected and repaired by DNA glycosidase. Okay, because, okay, take note, okay, because there will be the hydrolysis, spontaneous hydrolysis occurs at the end glycosidic bond. So in a glycosidase, enzymes helps to detect and repair the damage. Okay, at this specific bond. Okay. Okay, this represents on the statement okay, at the previous slides. Okay, so this is a removal on the purine. Okay, on the purine based by hydrolysis and glycosidic bond. Okay, and this one is the removal of amino group by hydrolysis. So it can happen. Okay, spontaneously, but cantik kan? It, it can happen, the damage can occur spontaneously, but there is a system to detect and repair it straight away. Okay, so yeah, so there are such enzymes that helps, okay, to detect and repair. Okay, okay, from spontaneous mutation, now we would like to talk about induced mutation. Okay, induced mutation meaning that there is other factor that led to the mutation. Okay, exposure to environmental agents can cause mutation. Okay, so for example, radiation. Okay, chemicals. Okay, UV. Okay, that's why now pun banyak kan lotion ke apa ke that has UV protection. Okay, they claim that it can protect from, it has SPF and SPF 50 lah apalah kan. So we would like to minimize, okay, minimize the this kind of agent, okay, to directly enter your skin, enter our system. We would like to minimize this kind of mutation to occur. So I think it's good to have that kind of creams, apa kan? It helps. Okay, it helps. We don't know at current our system, our planet now. I'm sure the UV that can be directly that reach to us may be at a harmful level. We don't know, so please be careful. Please just minimize, okay, minimize the exposure to this kind of mutation. Okay, bila masuk lab, if you are working in laboratory, please be careful. If you are working with chemical, first thing is please wear your gloves. Okay, kalau bekerja dengan chemical, gloves nombor satu, tak boleh tinggal. Okay, you don't know what kind of chemicals your friends are using. You don't know what kind of chemicals your colleagues are using. You don't know what kinds of chemical your seniors are working with. So you don't know what kind of chemical has been spilled. Okay, that been spilled in the bench. Okay, kita tak tahu. Okay, because some people dia tak label ke botol ke apa ke, ada spill kat lap ke apa, it can cause some problem to you. So be careful. Okay, so ionizing radiation is another agent that can if you exposed to in a significant time, significant period of time, it can lead to this kind of problem. Okay. All right. Okay. Free radicals on DNA and also in the process, okay, most cells can repair damage to low levels of radiation, which single-stranded damage easily repaired, but double-stranded more difficult, that can cause death of cancer. Okay, so take this information seriously. Okay, this is very important. So chemicals. So chemicals, okay, chemicals are also another mutagens that classified according to their mode of action. Okay, there are chemicals that the structure is heterocyclic, okay, which it can be incorporated into replicated DNA induced mutation by altering base pairing. Okay, so this type of chemicals, it can induce mutation. Okay, they alter base pairing. Okay, for example, 5-bromouracil is an analog of thymine. So it can be incorporated in DNA, which it can replace thymine. Okay, so bila dia kata analog, so be careful. So when you are working with this specific chemicals, just be careful. Okay. Okay, so this is 5-bromouracil, and it's analog to purine. Okay, reactive chemical mutagens. Okay, another one. Okay, reactive chemical mutagen. It can change the functional groups on the DNA base. Okay, for example, nitrous acid. Okay, okay. Ini ada di dalam lab. Okay, so second lab dia ada. So it can be aminate cytosine, uracil base pair with adenine. Okay, so okay, we don't expect this kind of damage can be caused by this chemical. Okay, and also alkylating agents are also mutagenic because it can add methyl or ethyl groups to base residues, and it can alter their base pairing characteristics. So these are the examples of chemical mutagens: nitrosamines, nitroguanidines, and alkyl sulfates. Okay. Okay, kita beri contoh lah. Alright. Okay, let's say cytosine can be, once it deaminated by this acid, nitrous acid, when it is deaminated, it becomes uracil. So adenine, it becomes hydroxyzine. So once bila ada mutation dekat sini, so your protein tu tak boleh nak function properly because it has been, the correct base pair has been replaced with other base. Okay, contoh satu sini lagi uracil, bila dia express, that your protein bila express dia dah tak functional. Okay. Okay, this is intercalating agents. Okay, so this is the hydrophobic aromatic chemicals that insert between stack base pairs in DNA. Okay, so it can cause mutation by insertion or deletion of bases, so increases the distance between bases. Okay, for example, ethylene bromide and ethylene orange, yang saya mention tadi. Okay, ethylene bromide. Ini guna kalau sesiapa yang ranjel ke apa ke, memang adalah ethylene bromide ni. Ethylene orange ni tak silap saya dalam dye, but I'm not sure. Maybe dalam dekat industri kan, ethylene orange. Okay, this dalam dye industry. Okay, so I'm not mistaken. Okay, so this is the structure of intercalating agents. Okay, it's ethylene bromide and ethylene orange. Okay, it can cause mutation by inserting or deleting, okay, base pair. Okay, so binding of intercalating agents to DNA can cause structural distortions. Okay, tengok ni terus, okay, the structure of DNA to distort. Okay, it change, okay, the structure of DNA, which led to lagi macam masalah. Okay, okay, another one, UV, okay, ultraviolet light. Okay, UV known to initiate chemical changes in DNA. Okay. Contoh dalam biasa, okay, I'm sure you biasa bekerja dengan apa tu, saya lupa lah, apa nama dia tu dalam lab tu. Okay, and before you can use that equipment, okay, apa nama dia, you have to switch on the UV light. Okay, we know that UV light meant to remove all bacteria kan, to make sure there is no bacteria or anything. But this UV light, you are exposed to it, it can initiate chemical changes in DNA. Okay, so how does this UV light can cause changes in DNA? It can cause covalent cross-linking of adjacent pyrimidine bases to form cyclobutane pyrimidine dimers. Okay, so it forms abnormal kink in DNA helix, which it leads to blocking of replication and transcription. So there will be no central dogma cannot occur continuously. DNA replication that will be transcribe that will translate because of the abnormal kink in DNA helix. Okay. And DNA photolayers can reverse dimer formation, and the deficiencies of DNA system can lead to skin cancer. So be careful if you are working with UV light. Dalam lab pun ada. Sometimes you need this UV light to before you guna apa nama dia tu? Saya tak ingat. Shall it work? Okay, be careful. Okay. Problems in DNA replication. Okay, the 3' overhang problem. Okay, 3' overhang problem, meaning that basically dekat lagging strand, the problem occurs in the lagging strand because we know that the addition of nucleotide occurs at the 3' end. So when there is an overhang, the overhang problem. Okay, so there will be no RNA polymer can be reconstructed in the normal manner. Okay, when the RNA polymer breaks down, we are left with an overhang at the 3' end of the translation chain and shortening of the transcript chain. Okay, so basically, dekat lagging strand, when we have this oxidative fragment, okay, we don't have any overhang of the 3' end. We hope that during the replication process, the 3' there will be no addition at the 3' end, so that there will be no overhang problem, because this overhang problem will shorten the transcript chain, so we don't have the translation protein, the protein that has been translated is not complete. Okay, so the lagging strand, the DNA polymerase needs to help to add new nucleotide to the 3' end, so that there will be no overhang. Okay, occurs at the 3' end. Okay. Okay, commutative replication problem. Okay. Okay, this one is the consequence of the end replication problem. Okay, this one has a. Okay, this one kalau ada overhang problem tadi lah, basically. Okay, bila ada problem and overhang problem. So this one, okay, this one juga when this terminal RNA primer cannot be replicated. Okay, terminal RNA primer cannot be replicated. Okay. Okay, so okay, this one. Okay, one strand replicates to the end. Okay, we have two strands. Okay, one strand, let's say leading strand has been replicates to the end. So the other strand, the other gap, okay, at the 5' end. Okay, so each chromosome in a cell that divides repeatedly will progressively shorten. Okay, so bila ada gap at the end of the replication, it will shorten. Okay, it will shorten the genes. Okay, so maybe some genes are lost. Okay, during lost from the end. Okay, bila ada problem dia akan shorten. So ada genes yang akan hilang. So what do you expect for the transcription and translation process of the genes? Okay, of the shortened genes. So that's the problem. Okay, ini overhang. This one. This is the overhang. Kalau nak tahu what is mean by overhang, bila ada overhang macam ni, dia akan problem lah. So we hope this overhang problem is not occurs when the addition of nucleotide berlaku dekat sini. Kita nak all this overhang protein. Bila ada overhang protein ni, dia akan shorten juga. Okay. Okay, how to overcome this problem? Okay, banyak lagi saya tak rasa sempat ni. Ada lagi few more, sikit je lagi. Okay. Telomeres and chromosome shortening. Okay, how to overcome this problem? Okay. Certain cell exhibit telomerase activity. Okay. This is telomerase. Okay, telomere. Okay, mesti tak dengar telomerase. Okay, so it's a very important enzyme. So basically, telomerase is combination of reverse transcriptase type enzyme and RNA strand that complement to the telomere sequence. Okay, so what does this enzyme do? So basically, it induces formation of the telomere DNA at the overhanging 3' end and adding nucleic acid to the 3' hydroxyl group. Okay, bila ada overhang DNA, telomerase comes in and it helps to add nucleic acid at the 3' hydroxyl group. Okay, so when enough telomeres have been added, primer is added, and DNA polymerase will transcribe the telomere strand similar to the action already described for autosomal fragment. So telomerase dia akan synthesize telomere DNA. Okay, so bila ada telomere DNA, dia akan muncul, lepas tu lah baru ada primer enzyme semua datang, so dia akan synthesize telomere strand tu kepada autosomal fragments. Okay, so what time is it now? Okay. How many minutes do we have? How many minutes left? Okay. Okay, I think I'm going to stop here on the telomeres. We are going to discuss on telomeres this Wednesday. Okay, and we are going to use the spare time nanti untuk revise balik on DNA replication before we move to transcription and translation later. Okay, ada soalan? Jadi saya stop dulu kat sini. So saya tak boleh habiskan kot. So I'm going to stop here at the telomere. We continue on Wednesday. Okay. So is there any questions?
Doktor,
Yes.
untuk tes Rabu ni, ada Zoom ke?
Kenapa kita ada tes tabu ni?
Nanti.
Oh, okay, test. If possible, saya memang nak you all guna Zoom. Boleh ke? Do you have any problem untuk guna Zoom untuk test? Okay kan?
Okay.
Alright. Okay. Do you have any other questions? Aiman? Hai Aiman. Yeah. Where's Aiman? Aiman dah tak ada dah? Kamalin Sofia, do you have Kamalin Sofia?
Yes, doktor.
Okay. Okay. Okay, how do you summarize today's topic? Do you want to share important points that you managed to get from today's lecture? Hmm? Share dulu. Okay. You nak buka kamera ke? Okay, in 5 minutes, let's summarize. Okay, let's summarize. What did you get from today's topic? Any important points? Okay, just share.
There are two types of mutations, spontaneous and induced mutation. Yeah.
Okay, thank you, Nasrul. What else? Apart from mutation? Tang Puimun.
Hello, Doctor.
Hi. Okay, any points you would like to share with your friends?
The DNA replication uses semi-conservative ways, and
Yes.
when replicating, it needs few enzymes to help in the process.
Okay, thank you. Thank you. Okay, can we have Marian? Marian Lee?
Yes, Doctor.
Anything that you would like to share? Yes, please.
It involves like 3 main processes during the replication, which is initiation, elongation, and termination.
Okay, thank you. Anyone would like to share on the enzymes involved in the replication process? How about Vivian? Okay, it's okay. Let's put up again on next Tuesday, Wednesday. Okay. So thank you very much. We are going to do some revision on Wednesday. All right. So thank you. Thank you very much.
All right. Thank you, Doctor.
Thank you,
Thank you,
Doctor.
Doctor.
Thank you, doctor.
Thank you, Doctor.